The binding of a hormone by its receptor tells us something about the nature of the hormone molecule. It suggests that the hormone molecule has a complementary region or a complementary conformation, no, that will be fitting to the region or a certain region of the receptor molecule. The degree of the similarity between these structures determines the tightness of the association between the hormone and the receptor. And this is said to be the affinity of the binding process. No? Aside from the biochemical nature of hormone receptors, we can also describe hormone receptors as uh, located in certain regions of the cell. There are hormone receptors found on the surfaces of cells. There are hormone receptors that are found inside cells. For the case of peptide and protein hormones, um, these hormones have receptors found on the surface of the cell, on the plasma membrane. And on the other hand, for the case of steroidal, retinoidal, and thyroidal hormones, these hormones cross the plasma membrane and look for their receptors within the cytoplasm, or receptors that can be found on the nucleus or can be found on the nuclear envelope. As proteins, it's important for us to delineate or to understand that hormone receptors are totally different from transport proteins that can also be found in certain in similar locations in the surface of the cytoplasm, no, or I mean in the surface of the cell membrane. Let's present a table describing the differences between hormone receptors and transport proteins. In terms of concentration, hormone receptors are very low. Okay, so in target cells containing receptors, receptors may be just 1,000 in number. The other hand, transport proteins within cells are very high. You know, in terms of binding affinity, receptors are specific. No, they're specific to the hormone that binds to them, while transport proteins, on the other hand, have low binding affinity. Some transport proteins can translocate uh, two or three types of molecules using the same passage or using the same structure. In terms of saturability, transport proteins are not saturable, while receptors are saturable. This is a very important feature that the hormone or the endocrine mechanism should maintain in order for it to be specific. In terms of reversibility, both proteinaceous structures, receptors, and transport proteins have reversible binding, have reversible interactions with molecules. And most importantly, the thing that delineates receptors from transfer proteins is the capacity of receptors to initiate signal transduction, to initiate the transmission of information, the transmission of signal that will activate intracellular functions as a result of hormone recognition and hormone availability in the bloodstream. Okay, now let's move on with our discussion by identifying different types of hormones. So hormones are classified into two general categories. There are hormones that are lipophilic, hormones belonging to group number 1, and hormones that are hydrophilic, those belonging to group number 2. Lipophilic hormones are hormones that are associated with plasma transport, meaning they can traverse the length of the plasma membrane of the cell membrane of the target cell. They localize themselves inside the cell, okay, and they look for their receptors in the cytoplasm or receptors on the nucleus or nuclear envelope, and then they activate a certain response, or they start a certain response in the cell. The other hand, hydrophilic hormones cannot cross plasma